Welcome to the first lecture in Unit 2, dealing specifically with membrane transport. As we talked at the tail end of Unit 1, we discussed membrane structure, and what we need to do is review that to make sure that those concepts are solidified so that we can move on from that foundation and talk about how molecules move into and out of the cell. So, if you recall from our previous lectures, we discussed the fact that the plasma membrane or the cell membrane is made up of a phospholipid bilayer. The phospholipids are a class of molecules known as lipids that have a hydrophilic portion and a hydrophobic portion. When we discussed the structure of the phospholipid, we talked about the fact that at the center of the molecule is a 3-carbon containing molecule known as glycerol. To 2 of those carbons, there is covalent linkages to fatty acid tails. This is the hydrophobic portion because the fatty acid tails are hydrocarbons, and these bonds that make up the hydrocarbons are nonpolar covalent bonds. Hence, these components of the molecule do not readily interact with water. The 3rd carbon in the glycerol is covalently linked to a phosphate group, which has an overall net negative charge, and this portion is the portion of the molecule that is hydrophilic. It is this dual nature of the molecule, the amphipathic nature of the molecule, that allows for the phospholipid bilayer to form. The hydrophilic heads of the phospholipids point towards the water, which would be on the exterior of the cell or the interior of the cell. And the hydrophobic tails point away or are tucked away from the water, as you see in panel A. Now, we also discussed the fact that the plasma membrane is made up or is recognized as what's referred to as a fluid mosaic model of membrane structure. That means that it is fluid, that it's not fixed and rigid. There's constant movement associated with it, and we'll review that on the next slide. It is also mosaic, which refers to proteins that are embedded in the phospholipid bilayer, similar to what you see in the diagram in figure B. So let's start by reviewing the fluid portion of the fluid mosaic model. What allows for the membrane to be fluid is the fact that each individual phospholipid is not physically linked to any other phospholipid. They are each individual entities or molecules in this phospholipid bilayer. So that alone allows for a certain amount of movement. Because these phospholipids are not physically linked to one another, there can be lateral movement, there can be rotation along an axis, and in rare circumstances, we can see the flip-flop, which is basically just moving from one layer in the phospholipid bilayer to the other. The biggest component that typically plays a role in fluidity of a membrane is the degree of saturation of the hydrocarbon tails or the fatty acid tails in the phospholipid structure. Saturated hydrocarbons or fatty acids have the maximum number of hydrogens bound to carbons possible, and this generates a very straight linear structure like you see on the right-hand side of panel B. This is because the straight linear structure allows for the molecules to come in extremely close proximity or contact to one another. The flip side of that is what you see on the left-hand side in panel B, which demonstrates unsaturated hydrocarbon tails. These unsaturated hydrocarbon tails are due to the fact that they do not have the maximum number of hydrogens bound to carbons, which means that there must be at least one position between 2 neighboring carbons that has a double covalent bond. What that means for the structure is that the structure is not linear. It has a bend or a kink in that tail. And when you have unsaturated hydrocarbons, this forces more space between neighboring phospholipids, and this allows for a much more fluid membrane. Lastly, cholesterol does play a role in fluidity in animal cells. When you have cholesterol, it typically makes your membranes a little more rigid, a little less fluid. But in lower temperatures, it actually hinders the solidification of the membrane, which would definitely be a really big benefit to an organism that is found at a lower temperature range, for example. So let's talk about the mosaic portion of the fluid mosaic model. The mosaic portion refers to the fact that proteins are definitely a part of every membrane. And the proteins themselves are typically found embedded within the phospholipid bilayer. So what you see here is something we discussed again in our previous lecture, and just categorizes some of the major groups of proteins that can be found associated with a membrane. There are those proteins that are involved in transport, which we will talk a little bit about today. Proteins can be involved in enzymatic activity. Oftentimes you have sort of an assembly line of proteins embedded in a membrane that have a similar function as far as a biological pathway is concerned. You can have cell membrane proteins be involved in signal transduction. That is, they can send or receive signals. They can be part of an intercellular junction, allowing for cells to interact and stick or adhere to one another. You can have cell recognition proteins that allow for recognition of one cell type versus the other. And then lastly, the broad category that we see on this slide are proteins that are considered anchors. And this allows for anchoring of the internal cytoskeleton, which we will talk about in our next lecture, as well as extracellular components can bind to these as well. So again, this is the mosaic portion. This is the portion where you have lots of different components in the phospholipid bilayer, and they are the protein categories we just talked about. Okay, so to round out the review of membrane structure, this is a figure we also looked at previously, and it's a really nice figure because it ties everything all together. What you can see in this complex figure is a plasma membrane or a cell membrane, the phospholipid bilayer, the proteins embedded in that phospholipid bilayer, and some of the functions of those proteins that we just talked about. And so again, what this demonstrates is how complex the membrane actually is. Okay, so now that we've gone through that review of membrane structure, what we want to talk about in today's lecture is the capability of the membrane to allow for exchange to occur. We need to allow nutrients to move in. We need to allow waste to move out, for example. And so how does this happen? This is because there are different properties associated with the membrane that we are going to discuss today. Now, what you can see in the very simplified figure that I have on the left is meant to be a phospholipid bilayer. And what you can see are some of the proteins that I mentioned just a minute ago that are considered transport proteins. So there are in fact some proteins that are involved in allowing for molecules to enter in or exit out of the cell. And what you can see from this very simple diagram is that some allow molecules to exit, some allow molecules to enter, and some can actually do both, have molecules enter and exit at the same time. Now, interestingly, what you can see on the right-hand side is a phenomenon that we're going to talk about here in just a minute, and that is that typically small water-soluble molecules can readily pass through the phospholipid bilayer. So this is again just an overview of a phospholipid bilayer, and molecules that are again small and hydrophobic tend to be able to pass readily straight through the phospholipid bilayer. They don't need any additional protein to transport them across, sort of just like parting the curtain and moving through. Small uncharged polar molecules can also move very easily across the membrane. However, large uncharged polar molecules, things like amino acids, glucose, nucleotides, cannot readily move past the membrane. And ions, like you see in the bottom of this figure, also cannot readily move. Now, we know that there's a need for amino acids, glucose, nucleotides, and certain ions within the cell. And so how exactly can those get moved into the cell if they cannot readily pass? And that's going to be, again, one of the focuses of today's lecture. One characteristic or feature of cell membranes is the term or the fact that they are selectively permeable or selective permeability. This is a property of biological membranes that allows some substances to cross and others to not be able to cross. It is selective in that only certain molecules can cross, and permeable basically means to permit. So selectively permeable membranes allow for certain molecules to cross and others not to cross. And that was sort of highlighted in the last figure that we looked at on the previous slide. So then what that transitions us into is beginning to discuss how exactly molecules can pass into or out of the cell. There are two broad categories of movement. There is what's referred to as passive transport and active transport. Passive transport is one that does not require any additional input of energy for the movement to occur, whereas active transport is going to require the input of energy to allow for the movement to occur. So two broad categories or two different ways that molecules can move across. They can either move across in a passive way or an active way. Passive transport, again, does not require the input of energy. Active transport does require the input of energy. So first we'll talk about passive transport, and the first mechanism of passive transport is referred to as diffusion. Diffusion is a physical process in which molecules move from an area of high concentration to an area of low concentration. How does this movement occur? It's due to the intrinsic kinetic energy of molecules. Molecules are always in a constant state of motion. And because of the fact that they're in this constant state of motion, this is what's going to allow for the property of diffusion to take place. In this very simple example that you see on the slide, if you focus on the left-hand side, what you have in this situation down the center of that figure in the yellow dashed line is meant to represent a membrane. And in this case, we have molecules labeled as dye, but the molecules can be anything in this scenario, on one side of the membrane, but not on the other. In this case, the membrane is permeable to the red molecule, means it will allow the red molecule to pass, hence the dashes that you see in the yellow membrane on that figure. What happens with diffusion is, again, molecules move from an area of high concentration to an area of low concentration. So in the far left and on the left-hand side of that figure, you have a high concentration of the red molecule. What happens is these molecules begin to bump and bounce around into one another, and eventually some will bounce across the membrane to the other side where there is a lower concentration of the red molecule. In the center of the figure, you see diffusion taking place, and basically it is a two-way street. Molecules can move one way and the other in diffusion. However, the net movement is always going to be from the high concentration to the low concentration until you get to the far right-hand figure, which is a state of equilibrium. In an equilibrium, you have roughly the same concentration of the red molecule on either side of the membrane. Do the molecules still move? Yes, they do. But roughly, you have the same concentration on either side of the membrane. So just to recap, one way or one mechanism that molecules can move across the membrane is by simple diffusion. And the only way this is going to happen is if the membrane is permeable to the molecule in question, and you have a concentration gradient where there's a higher concentration of the molecule in question on one side of the membrane compared to the other. And that is when you will have diffusion take place. So now we move on to talking about the passive transport of water. This is the diffusion of water, and it is referred to as osmosis. So osmosis is specifically the passive transport of water across a membrane. And it is a really important concept because we know how important water is to cells and to life as we know it. So before we talk about osmosis and the examples that you see on the slide, I'm going to introduce a series of terms. When we talk about osmosis, we will always be comparing two different solutions. When we compare the two different solutions, we can have terminology associated with those two solutions. So I'm going to start off with an isotonic solution. And an isotonic solution is going to be where we're comparing two solutions that have the same solute concentration on either side of the membrane. The other two terms are hypertonic and hypotonic. The hypertonic solution, when comparing two solutions, is a solution with a greater solute concentration, and the hypotonic solution is going to be the solution with the lower solute concentration. So when we're talking about osmosis, we're not only referring to water movement, but we also have to have some understanding of the solute in question and the solute concentration. Now, in this case, water is permeable to the membrane, but the solute usually involved in these situations is not, meaning it cannot pass readily through the membrane. So again, when you compare two solutions, you're going to use the term hyper and hypotonic together. One solution will be defined as the hypertonic solution, then by default, the other one you're comparing it to has to be the hypotonic, and vice versa. If you've identified a solution on one side of the membrane as hypotonic, the one on the other side has to be hypertonic. The exception to that is when we're dealing with isotonic solutions, and those have the same solute concentration on either side of the membrane. So this figure highlights what happens in certain situations when you place cells into different types of solutions. Now there are two sets of figures for a typical animal cell and for a typical plant cell. We haven't discussed cellular structure in detail yet at all, but there's a clear defining characteristic of plant cells, and that is that plant cells have a cell wall on the outside of the plasma membrane. This allows structure and protection and rigidity to the plant cell. But what that means is that it is a physiological barrier that basically can't be passed. And so that's going to become important when we talk about what's happening with these cells on this slide. All right, so let's look at panel A. Panel A in a isotonic solution is going to be one where the solution on the outside of the cell and the inside of the cell have the same solute concentration. Water, again, in this example of osmosis, can readily move across the membrane. And so what you're going to have in both cases is water movement in and out of the cell, basically at equilibrium, because we have the same solute concentration inside and outside. Okay, so that's the normal situation for the typical animal cell. And actually for plants, it turns out that this is what's referred to as flaccid, where the plant cell is actually not completely filled with fluid or filled to the brim with fluid, and you have a plant that sort of looks a little bit wilted. We'll explain that in just a minute, a little more detail. In panel B, what you see is a situation where if you place the cell in question into a hypotonic solution, that is a solution with a lower solute concentration, water is going to move into the cells in both of the circumstances, both animal and plant example. This is because the hypotonic solution that you placed your cells into has a lower solute concentration than the inside of the plant. Remember, the solute cannot move, but water can. So now there is a difference in water concentration on either side of the membrane. And remember, when we talked about diffusion, water will move from an area of higher concentration to an area of lower concentration. So when you place either the animal cell or the plant cell into a hypotonic solution, water is going to move into the cell. For an animal cell, typically, depending on the concentration of the solution, water will move in and eventually cause the cell to burst or lyse. Basically kills the cell because water has blown up the cell in question. For a plant cell, because there is that physical barrier, the cell is not going to blow up or lyse or explode, and that's because, again, there's a physical barrier. And this is what is referred to as the Detergent State, and this is the state that most plant cells are in a normal, everyday setting. In panel C, what you're looking at is a situation where you would place a cell into a hypertonic solution. In a hypertonic solution, the solution is going to have a greater solute concentration than the inside of the cell. Remember, the solute cannot move, but water can. So again, water will move from an area of higher concentration to an area of lower concentration and moves out of the cell. And in this case, you do get a shriveled cell that can actually be identified under a light microscope. Now, the last type of passive transport mechanism I want to discuss is something referred to as facilitated diffusion. And basically, it's what its name implies, is that there is some facilitation or some help that's going to occur. The mechanism, however, is still a mechanism of diffusion. Molecules moving from an area of high concentration to an area of low concentration. However, with facilitated diffusion, there is a molecule that is usually referred to as a channel protein or a carrier protein that allows for the molecule in question to pass through the membrane. So again, facilitated diffusion just means that there is some additional help, but the help is just in the form of forming a tunnel or a channel across the membrane. Diffusion is still the main concept that allows for this movement to occur. Okay, so let's transition now into active transport. Active transport, as we defined previously, is a mechanism of transport that requires the input of additional energy to allow molecules to move across the membrane. Interestingly, though, with active transport, you do have a protein involved that is referred to as the carrier protein or transport protein. And what you also have is a conformation shape change that usually occurs in the transport protein to allow for the transport of the molecule across the membrane. And lastly, with active transport, you are moving against the concentration gradient. So with diffusion, again, you move from an area of high concentration to an area of low concentration. Active transport is going to be a situation where you move against the concentration gradient from an area of low concentration to the area of high concentration. Because you're moving against the concentration gradient, this is why there is the requirement for an input of energy to allow that to occur. So in the figure, what you're seeing is that energy in the form of ATP, which hopefully you are familiar with the fact that ATP is the main energy source for the cell, and we will talk about that in our next unit, or actually at the tail end of this unit. But for now, just know that that is the energy molecule that provides the energy for the movement to occur, and that movement is usually due to a change in the shape of the molecule that is transporting, or the protein that is transporting the molecule in question. So now let's look at a specific mechanism of active transport. This specific example of active transport is with what's referred to as a sodium-potassium pump. And a sodium-potassium pump is a membrane-associated protein that basically does just what the name implies, is move sodium and potassium across the membrane. In the top right-hand figure, you're looking at an overview of what the sodium-potassium pump does. And Essentially, it allows for sodium to be moved out of the cell and potassium to be moved into the cell. Both are going against their concentration gradient. And so what you need is a mechanism that imports or utilizes energy to allow for the movement of these two to occur. In the left-hand panel, what you're looking at is sort of a sequence of events that takes place during the transport mechanism. So what I want you to do first is look at the figure on the left-hand side. Sodium is being represented by the circles, and potassium is being represented by the squares or the diamond shape. If you look at the 11 o'clock position on this figure, what you see is sodium entering into the transport protein into slots or divots that allow for the sodium to bind. Next, at the 12 o'clock position, what you see is the little energy burst from ATP that is going to allow for a conformation change to occur. Now, we're going to talk about how ATP contributes energy to reactions, but in this figure you can see what's happening is a phosphate group from ATP is being transferred to the transport protein, indicated by the really tiny letter P in a circle. And what this does is causes a conformation change in the transport protein. So then if you transition to the 2 o'clock position, you see that conformation change has changed the shape of the protein to where it's no longer pointing open towards the inside of the cell. It is now pointing open towards the outside of the cell. Also, the places where sodium has bound has physically changed too, which basically causes the sodium to be pushed out of the slots or the spaces and now exits the cell. Once that conformation change has occurred and the sodium is released, that then allows for the potassium to bind into the potassium slots. And that's sort of at the 4 o'clock position. When potassium binds, that triggers the release of the phosphate group. And once you have the release of the phosphate group, like you see at the 6 o'clock position, the conformation change occurs again in that transport protein to where now it's no longer pointing outside of the cell, it's now pointing open inside of the cell. And this now again allows for the potassium to enter into the cell. And then this cycle continues over and over and over again. But what this demonstrates is a mechanism of co-transport, meaning two different components are being pumped across, in this case ions, sodium being pumped out, potassium being pumped in. Both are going against their concentration gradient, hence the requirement for the input of energy to allow for this movement to take place. So another mechanism of membrane transport is exemplified in this particular slide. But before we talk about this type of membrane transport, which again is still a mechanism of active transport, just more of a specialized mechanism of active transport, I want to define a couple of terms for you. The first one is that all cells have a voltage across their membrane. And a voltage is just a separation of separation of opposite charges. And here in this case, the voltage is generated by the plasma membrane. This is referred to as a membrane potential. So specifically, the voltage generated by the membrane, where you have a separation of opposite charges, is called the membrane potential. Typically, the outside of the cell has a net positive charge, and the inside, just inside, has a net negative charge. Now, because of that, ions may actually have a hard time being moved across the membrane because not only do you have the chemical composition that needs to be addressed, basically overcoming the chemical gradient, what exactly is the molecule or ion in question, But you also have to deal now with an electrical gradient, that there's this difference in charge that occurs. And so the combination of the two makes it even harder for certain components, certain ions, to get moved across the membrane. This is overcoming what's known as the electrochemical gradient. Not only what it is, but what charge it is, makes it dually difficult for certain ions to move across the membrane. So in that case, you do have some specialized membrane transport proteins, like you see in the bottom left-hand corner. This is what's referred to as a proton pump, and a proton pump does exactly what its name implies, is that it pumps protons across the membrane. You can see on the left-hand side of the figure, in the beige color, is the inside of the cell, the cytoplasm, and on the right-hand side of that figure in the blue is the extracellular component. Because there is this difference in charge across the membrane, and you also have a higher concentration of hydrogen ions outside of the cell in this case compared to the inside, this makes it again dually hard to get hydrogen ions across the membrane. And so this is another example where you would need a transport protein that utilizes active transport, the energy associated with ATP, to get the movement of a particular component across, in this case a proton. Okay, so let's review really quickly the transport mechanisms we've just discussed before we continue on. And that is, we've discussed the category of passive transport as well as active transport. Passive transport, by definition, does not require the input of additional energy to allow for the movement to take place. It is based off of simple diffusion, that is movement from an area of high concentration to an area of low concentration. So on the far left is simple diffusion, where the molecule would just simply pass through the membrane. In the center is facilitated diffusion, where again you have a concentration gradient established, but there is still a channel protein, a tunnel protein, that allows for the movement of the molecule in question. And then lastly is the example of osmosis, which is the movement of water from an area of high concentration of water to an area of lower concentration of water. But in order to understand those mechanisms, you have to understand the solute concentration in question. And then we also discussed active transport, which requires the input of energy, requires a transport protein, as well as, in this situation, the molecule in question is moving against the concentration gradient from an area of low concentration to an area of high concentration. Now, I include this slide. It's a very simplified view of a cell, but it's really just a good reminder for me to discuss the fact that every cell is going to have characteristic sets of membrane proteins, depending upon what the cell's function is, that type of thing. And so you can see that you have these variety of transport proteins associated with the plasma membrane of the cell. In addition, though, when we're talking about cells that are eukaryotic, which again are cells that have organelles, and we're going to talk about those in more detail in our next lecture, these structures inside the cell are membrane-composed structures, and these components also do typically have membrane-associated proteins. And so again, it's just a nice way for me to make sure that I recognize and acknowledge and let you know that we're not only talking about membrane-associated proteins that are plasma membrane-associated, but also membrane proteins that can be found on internal organelles inside the cell. Now, the last broad category of movement that I want to discuss across a membrane deals with molecules that are relatively large. So everything we've discussed to this point are still relatively small molecules, whether they move freely across the membrane or require a protein to help them move, they are still relatively small molecules. There are situations, however, where a large quantity or large size needs to be moved into and out of the cell. The general mechanisms for that are going to be listed on this slide and on the next slide. So exocytosis is a mechanism of secretion of large molecules utilizing vesicles. So here is a mechanism to allow for large molecules to exit the cell, exocytosis. And what happens in this mechanism is that you have a structure inside the cell that is referred to as a vesicle. Now, a vesicle is just defined as a small membrane-bound sac. So it is a membrane-associated small structure. And in this case, we've got this purple molecule that needs to leave the cell. It's in a vesicle, which is this small membrane sac. And what happens is the vesicle moves towards the plasma membrane and eventually fuses with the plasma membrane. So this goes back to that fluidity that we talked about is so important in understanding how movement can occur into and out of the cell, is that the fluidity allows for the fusion between the plasma membrane and the vesicle membrane. Once that fusion takes place, the contents of the vesicle can now be forced out of the cell, like you see in the far right of the figure. So the opposite of that is a mechanism known as endocytosis. This is where large molecules are typically taken into the cell. And the mechanism is basically taking that mechanism we just looked at and putting it in reverse. Starting on the left-hand side of the figure, you see a little dip or divot in the plasma membrane, and you see these green molecules sort of fitting in that dip or divot or pit. Eventually, the membrane will close up on itself, like you see in the center of the figure. And lastly, it will pinch off from the plasma membrane, forming a vesicle with the green molecule contents inside. Now this molecule is inside of the cell and can be transported to whatever location the cell needs this particular green molecule. Now there are in fact some specific mechanisms of endocytosis found in animal cells. The first example is a mechanism known as phagocytosis, and the word phago comes from the Latin phagin, which means to eat. So typically with phagocytosis, you have large molecules that are being taken in a sense being eaten if a cell could actually eat that molecule. So usually it is a food or a fuel source. However, another example might be cells that are part of the immune system. Many cells that are part of the immune system are phagocytes. That means they are cells that participate in extreme amounts of phagocytosis. What those cells are doing is taking in and entrapping pathogens, typically bacterial cells, and destroying them overall. Pinocytosis is the next example, and the root pino comes from the Latin meaning to drink. So here what's typically occurring is an impinging of the membrane where you have extracellular fluid and of course any solutes that are dissolved in that extra fluid coming into the cell. And then lastly, you have receptor-mediated endocytosis. And this is movement of specific molecules into the cell that involves the inward putting of these vesicles that contain proteins with receptors that specifically bind to the molecule in question. And that sort of rounds out our discussion of transport into and out of the cell.